Hi, I'm Jabran Shabib, and I'm an evolutionary geneticist at the University of Edinburgh. I'm currently a European Research Council postdoctoral fellow in Peter Keeley's lab, where I'm trying to answer questions about the prevalence and distribution of mutations. Here I'm going to talk about the work we did using whole genome sequences from mice of 4 different lab strains. An inbred mouse line is a population of mice that have been maintained exclusively by brother-sister mating, also known as full sim mating, for many generations. This reduces genetic variation within lines to close to zero. Genetic uniformity is really the key goal of an inbreeding program. It is important because inbred strains allow experimentals to vary only the parameters of interest and to measure their effects, ruling out genetic variance, which in turn is important for estimating relative contribution of heredity and environment, the relative importance of different environmental factors on traits of interest, and for discovering the phenotypic effects of mutations. It is also important for experimental reproducibility and comparability, because you want to be able to say that the results observed in one lab in individuals from a particular strain are also applicable in any other lab using individuals from that same strain. Inbred mice lines are typically maintained by brother-sister mating for many, often hundreds of generations. A number of companies maintain these inbred lines, but they originally come from the same stock. These companies have a nucleus colony from which they create the next generation of mice for breeding, expansion, and production colonies. These expansion and production colonies are not fully inbred, but allow these companies to meet the demand of the research labs ordering them. We used Illumina technology to sequence 4 pairs of mice with high sequence coverage. 1 pair of mice came directly from the nucleus, and the 3 other pairs came from production colonies. We looked to add single nucleotide polymorphisms that were present in one strain and absent from all the others. These represent the variants that have recently arisen as new mutations in a given strain. Because of the fact that there's only one breeding pair per line, and what we know about mutation rate per nucleotide site in mice, we could get a theoretical prediction of how much genetic variation to expect at mutation drift equilibrium. This was done through simulation with an arbitrary mutation rate of 10 to the negative 8 per site. I say arbitrary because the relative proportion of elements in the site frequency spectrum is independent of mutation rate. The number of observed variants in the sequences for each pair of mice was then compared to the simulated site frequency spectrum, and an optimization algorithm was implemented to find the mutation rate that would minimize the differences between the observed and simulated site frequency spectrum numbers. What we found was that in 3 inbred pairs of mice obtained from the production colonies, there was far more variation than expected. This translates into higher than predicted mutation rates, whereas in the pair directly obtained from the colony nucleus, mutation rates closely match the theoretical expectations and estimates from previous studies. We believe that this difference is due to de novo mutations that arose in the generations between the nucleus and the production colony, which could be up to 10 generations. Working with inbred mice typically assumes genetic uniformity with strains, but this is not the case. Individual mice will differ at many hundreds of nucleotide sites across their genome, which could be affecting the traits of interest. Therefore, we suggest caution when interpreting the results of experiments using mice from production lines, especially regarding the reproducibility and comparability to other experiments that use the same inbred lines. Therefore, if you're working on inbred strains, it's important to specify the origins of your strains, including where the mice came from and any substrate information you might have. If you're breeding the mice, well, then keep good records of your pedigrees. Thank you.